The last video cut off right before the last but largest lymphatic organ, and that's the spleen. It's made up of two types of tissue, red pulp that has sinuses filled with red blood cells, and white pulp with lymphocytes, macrophages that are surrounding branches of the splenic artery. The function of the spleen is blood production in the fetus, a blood reservoir, a red blood cell disposal, and of course monitoring blood for foreign antigens. The spleen is highly vascular, and so it is vulnerable to trauma and infection. If it's ruptured, that is going to require its removal, which is a splenectomy. And there it is. Okay, so the bulk of this video is going to focus on resistance, resistance to pathogens. So let's define pathogen before we get too far. Pathogens are environmental agents capable of producing disease. So we can be talking about quite a few different things, but we'll just leave it at pathogen for now. And so our three lines of defense, we're going to go into more detail, but first would be our external barriers, our skin and those mucous membranes. The second is our non-specific defense mechanisms, and then third is the immune system. So here we'll focus on the first two. So non-specific resistance means we're guarding against a broad range of pathogens, so not being specific, and so the effectiveness of this guard doesn't depend on prior exposure to a specific pathogen. It basically means we're just going to keep out everybody, not being specific to a certain type of pathogen. So we were going to talk about the skin first, but then also as we get deeper into the body, leukocytes, macrophages, and then immunity would become a specific resistance. And we'll get to that in the next video. So let's focus on our first line, and that is our external barrier, the skin. We've talked about this in A&P1 with the integumentary system, but the skin is a mechanical barrier. So it physically protects pathogens from entering the body. The toughness of the keratin makes penetration difficult. It is dry and nutrient poor, so it doesn't support microbes growing, and then those dead skins coming off. If anything lands on there, then those outer skins are exfoliated soon, hoping to remove anything. So the acid mantle is also part of the skin's protection because it is a little thin layer of lactic acid, and so again also inhibits bacterial growth. Also, along with the skin, we have the mucous membranes in all of our openings to the exterior of the body, so digestive, respiratory, urinary, and reproductive tracts. They're physically trapping those microbes. And then in the mucus, there's enzymes, and that is breaking down the cell walls of the bacterial cells in hopes to destroy them. Okay, so then our next line of defense are phagocytes. Those are cells that are using phagocytosis to ingest those foreign particles and destroy them. And so our 5 types of white blood cells, which you've gone through previously in the blood chapter, but in this chapter we're going to focus a little bit more about each of their functions. So the neutrophils specifically are going to move through the connective tissue, killing bacteria by phagocytosis. Again, that's an active transport where the cell is able to ingest the foreign cell and break it apart and kill it. It also talks about, in the book, this is going into a lot more detail, but it makes kind of a killing zone, and those lysosomes discharge enzymes, and they push the enzymes out into the fluid, and it basically makes an area that is going to kill the bacteria even before they're ingested in the cell. The eosinophils are found especially in the mucous membranes. They are active specifically against parasites and allergens. And so they're focusing on killing tapeworms, roundworms. They produce these toxic substances, and they also are able to use phagocytosis against antigens and antibodies. So from the blood chapter, you know that an antibody is going to attach to an antigen, and when those complexes form, the eosinophils are able to break them apart and kill them. Mostly here you want to focus on parasites and allergens. So eosinophils are able to limit the action of histamine and other inflammatory chemicals. Our third type are basophils. They secrete chemicals that help to move and get the action of the other leukocytes to occur. And so specifically here we're talking about leukotrienes that are activating and attracting neutrophils and eosinophils, and then secreting histamine to dilate the blood vessels to increase blood flow. That helps new leukocytes arrive to the area where the pathogen is, and then heparin, which inhibits clot formation. A clot would slow down the leukocytes getting to the site of the pathogen. Okay, we're going to talk more about lymphocytes in the next few videos, but we'll talk about them briefly here. So lymphocytes, we've already introduced T cells, B cells, and those NK cells, but T cells being the most prominent. They have lots of diverse functions. We will get into them more when we get into the immunity, but they are specific immunity against certain pathogens. And our last type is monocytes. They are emigrating from the blood and moving towards the connective tissue. And when they are there, they transform into macrophages. So the macrophages are containing all of the body's phagocytic cells except leukocytes. So they are basically hunting down these pathogens, ready to kind of consume them and break them apart. Okay, the second line of defense also includes proteins, proteins that inhibit microbial reproduction. They provide a short-term, non-specific resistance to bacteria and viruses. And two families of proteins are also included in our second line of defense. Interferons are secreted by certain cells infected by viruses. And so they don't really help the cell that secretes them, but they tell neighboring cells and protect them from becoming infected. So we know that the virus soon is going to be released from that infected cell and would potentially be ready to infect neighboring cells. So the interferons kind of alert them, and it binds to surface receptors on the neighboring cells. Continuing with the interferons function, that alerted cell is then going to make proteins that defend from infection, and it also activates NK cells and macrophages, and then those NK cells will be able to destroy the malignant cells. And then the last part of the second line of defense is the complement system, and that's a group of 30 or more proteins that we're going to see as part of this non-specific resistance, but also part of the next video about specific immunity. They're synthesized mainly by the liver. They circulate in the blood in an inactive form, but then in the presence of the pathogen, they're activated. So this goes into kind of different pathways of the complement activation, but all three of them result in the protein group being activated, and they, when activated, are going to have 4 methods of destroying the pathogen. So this focuses on the different pathways. There's 3. We have a specific immunity, which we're going to get to in the next video, but a specific immunity would have a classical pathway. But then let's here, since we're still on the non-specific resistance, let's look at the alternative and the lectin pathway. So those are both non-specific, again, in general for any type of pathogen. And this talks about triggering a cascade reaction. And so this pathway, I would encourage you to read more about it in your book, but it is extremely specific, and reactions triggering reactions. And so we're going to not go into so much detail because it is a little overwhelming. We're just going to jump to the point of the complement system being activated, and there's 4 mechanisms, 4 ways that the pathogen is destructed. So one is inflammation. C3A is what stimulates those mast cells and basophils, and they're secreting histamine and other chemicals, and so that then kind of sends a signal to the neutrophils and macrophages that it's time to come, and that helps to speed up the destruction of the pathogens. Another mechanism is immune clearance, where the C3b binds to the antigen-antibody complexes on red blood cells, which we already talked about in the previous chapters. And so macrophages in the liver and spleen destroy those complexes, and so after that has occurred, then those red blood cells are unharmed. So it basically clears away those foreign antigens from the bloodstream. The third type or mechanism is phagocytosis. The neutrophils and macrophages can't use phagocytosis on naked bacteria, viruses, or pathogens, so they cannot identify those cells if they are not able to identify them. And then cytolysis is where the complement system attracts more proteins, and they make what's called a membrane attack complex. So it's a hole in the target cell, and basically the ions are going to come out, the water flows in, and then the cell will rupture. Again, I would encourage you to read over this in your textbook, but you can see that there's lots of reactions that are going to take place, and again, the pathway is all 4 of those mechanisms, or the 3 different pathways lead to the 4 different mechanisms of destroying pathogens.